12th Bio-botany Chapter 4 Principles and Process of Biotechnology. In Part 12, we shall discuss about RNA interference, RNAi. We have already discussed about gene expression. When a gene is expressed, its protein product will be obtained. All characters of organism are due to expression of different genes. Gene expression takes place by means of transcription and translation. What is transcription? One of the strands of DNA is copied into mRNA that takes place inside the nucleus. When the mRNA is transcribed, it has introns and exons. When the introns are spliced out, removed, a functional mRNA will be formed. It has only exons and 5' capping and tailing at 3' end. This functional mRNA is exported to the cytoplasm, where it combines with ribosomes and form protein when amino acids are connected by peptide bonds. This amino acid chain undergoes folding to form a functional protein. This is usual mechanism. We have already discussed about gene regulation in this lesson. We discussed about gene regulation or gene expression in zoology. Here there is another pathway to control gene expression, that is RNAi pathway, RNA interference pathway. RNA interference is a biological process in which RNA molecules inhibit gene expression or translation. What is transcription? DNA is copied into mRNA. mRNA is translated into protein. When the RNA is silenced, then no protein will be formed. So translation is stopped by inhibiting RNA functioning. This is done by neutralizing targeted mRNA molecules. Normally mRNA is translated to protein, then mRNA is neutralized or silenced. In this way, gene expression is stopped. A simplified model for the RNAi pathway is based on two steps, each involving ribonuclease, that is RNase enzyme. RNase enzyme or ribonuclease, it cuts RNA molecule. In step 1, trigger RNA is processed. Trigger RNA is either double-stranded RNA or microRNA, primary transcript. Before processing, it is called a primary transcript. The microRNA is a small RNA molecule containing about 22 nucleotides. The microRNA or double-stranded RNA or the trigger RNAs. The trigger RNA is processed. After processing, short interfering RNA, siRNA, is obtained. Trigger RNA is processed into siRNA, short interfering RNA, in step 1. This process takes place by enzymes Dicer and Drosha. They are RNA's second categories. They act as molecular scissors. This is double-stranded RNA. This is short hairpin RNA. This is artificially developed RNA. Trigger RNAs are cut into small pieces, that is small interfering RNA by Dicer or Drosha. This is step 1. In step 2, the short interfering RNA, siRNA, combines with a complex. This is short interfering RNA. siRNAs are loaded into the effector complex. In step 2, siRNAs are loaded into the effector complex. What is effector complex? RISC complex. RNA induced silencing complex. RISC, RNA induced silencing complex. In that complex, the siRNA is converted into single stranded RNA. The double stranded siRNA is converted into single stranded RNA. RISC complex incorporates one strand of small interfering RNA. The small interfering RNA is converted into single stranded RNA. So only one strand combines with RISC complex. This is RISC complex. The brown color is one of the strands of siRNA. The green color is target mRNA. The siRNA pairs with the complementary nucleotides of target mRNA. After pairing of the siRNA and target mRNA, the RISC complex enzymes cut the mRNA. So mRNA is inactivated. The siRNA is unwound. So it becomes single stranded during RISC assembly, and the single stranded RNA hybridizes with mRNA target. This is mRNA target, where hybridization takes place between complementary nucleotides of siRNA and mRNA. This is siRNA. siRNA becomes single stranded. This is mRNA. Both hybridize. After hybridization, the nuclease enzyme cuts. RNase enzyme cleaves the RNA. So RNA becomes inactivated. No protein is produced. This RNAi is seen in plant feeding nematodes. Nematodes are called as roundworms. They damage to the host plants. They cause damage to the host plants. This is plant cell. The plant cell has double-stranded RNA. It produces double-stranded RNA. It combines with dicer, and because of the interaction of dicer, siRNAs are produced. Small interfering RNAs are produced. When the nematode eats the root of the plant cells, the siRNAs enter into the digestive system. Here the interesting thing is that the siRNAs enter into the wall of the digestive system and enter into the body of the nematode. And inside the nematode, the RISC complex is unzipped. RISC complex opens, and siRNA becomes single stranded. It combines with target mRNA. The target mRNA is cut into pieces. So the mRNA is inactivated. It becomes silenced. The protein product is not produced. Because of this mechanism developed in plants, the nematode cannot cause any damage to the plant. In this way, the plant is protected from nematode without any insecticides.